about technological advances in accelerated wound repair, keloids, scars, stretch marks, and facial rejuvenation. Technological advances in accelerated wound repair and regeneration is actually a study that I conducted along with Dr. Lampe, and there is a publication that is associated with this. So this presentation is based on this publication. The skin responds to injury with a series of dynamic processes: coagulation, inflammation, tissue formation, and remodeling. Inflammation is present in adult but not embryonic wounds that heal without a scar. And this is a study that I'm basically enclosing here. Depletion of one or more of the inflammatory cell lineages enhances wound healing, and chronic and persistent inflammation is basically associated with most chronic wounds. Persistent inflammation underlies abnormal healing, and that is basically what all of the studies demonstrate. Dysregulated or overly exhibited signaling pathways underlie abnormal healing. Basically, when the signaling pathways, the signaling processes recruit neutrophils to kill invasive microorganisms and microfibers to clear apoptotic cells and develop early wound closure. Now, what happens after that? The mast cells release histamine and other substances that ultimately result in scar formation. Entirely normal healing is observed at the genetic depletion of mast cells. So signaling is also looks at the chemokines and inflammation. Signaling also is involved. All of these elements are involved in how a scar will heal. And whether that scar will become a keloid or hypertrophic scar that are basically based on dysregulated signaling, because dysregulated signaling will cause aberrant wound activity. Hypertrophic and keloid scars are pathological scars. They result from aberrant inflammation and overactivity of wound fibroblasts. Basically, the body is panicking and overreacts. It's an overreaction, it's a panic response. This aberrant wound activity reflects dysregulated signaling that is not switched off, but continues. And that is, again, healing is important. But when the healing keeps going, after it should stop, then you have this problem with keloid and hypertrophic scars because it continues aimlessly. It's not directed. Laser efficiency on wound keloids and hypertrophic scars, they have had reported efficiency, and their main type of lasers, ablative lasers, remove keloids, and they're very useful in that area. And non-ablative lasers basically induce necrosis, but without removing the keloid. There are scientific issues on several applications. Laser studies report limited or no follow-up, however, and side effects of ulceration or hyperpigmentation are also side effects that are unwanted results, and only 66 to 77% of patients experience greater flattening of the scar, with 21 to 35% having a recurrence rate. And that's why when you need to have a longitudinal study to see what will happen after 1 year, are you going to get hyperpigmentation? Are you going to get the same thing coming up again? So laser efficiency of wound keloids and hypertrophic scars, there has some research issues as well. Statistical significance is not convincing when the results are subjected to critical analysis, because you look at, let's say, 0.0001, but then when you look at the people, 8 out of 22 people, for example, had a clear reduction in the size of their lesions, but 10 of them had a slight reduction. So, if you look at it like that, then you just measure both the slight reduction and the results will be significant because they're towards the right direction. However, it does not mean that the treatment is necessarily effective. So here is an example of statistical significance that you have to look at the detail and not just the average. So endogenous electrical fields in wound healing and ultra-low energy devices are very, very real. There's a lot of research on that. Endogenous, called endogenous ionic currents with DC electric fields. There's a lot of research. There's just many articles on that. And the endogenous ionic currents or electrical fields arise instantaneously in the skin during healing. Basically, it's something that the body does to heal. And if you do it, of course, from the outside and you enforce that process, you will get a much better endogenous fields, and therefore you will basically help to control the wound and increase the healing. Also, there's a lot of studies that for microcurrent stimulation that have that report that there is a signaling and DGF-1 B1 release in fibroblasts and osteoblasts like cell lines. And there is a lot of, this is the research that shows how the healing was significantly improved with this particular technology. Research with ionic currents has reported enhancing soft tissue healing, increased secretion of growth factors, and adenosine ATP production, adenosine triphosphate ATP. And this is the studies that involve bioelectricity, and this is some of the results that these studies have demonstrated. Again, there is a vast literature showing that. And then there is the other technology with ultra-low microcurrents that, again, there's no signaling, there's a digital technology, but they look at the management of diabetic wounds and hypertension and chronic wounds, all the wounds that, are very important to treat, because if you don't treat, these people can get the legs amputated. So it's quite significant, these results. And a lot of these results are from Lee et al., and they used a ultramicrocurrent device that has a continuous pulse and goes all the way down to 400 nanoamperes. Now there's even further, there's more technology, that's the technology that we use. That is ultra-low energies. And what we have found is that in these very ultra-low energies, there is a book that was published, electron-gated ion channels. What happens is that if that energy is below thermal noise, for some reason that we don't know why, the cells the ion channels of the cells open up. And then you can insert different signals that will, this is the book, Electron-Gated Ion Channels, and it has a lot of mathematical calculations on how that happens. And basically what's going to happen is that you can insert signals. There's also something very interesting that happens at molecular mechanisms. They follow the rules of quantum mechanics, which are very different than any other laws in physics. And basically what you see is, for example, if you change one electron, by donating one electron, you make a molecule go back to its stable state. So there is sort of a time reversal, and that comes with ATP that turns into ADP, or ADP that turns back into ATP. It gets reversed kind of routinely. It happens with GAP, with with GTP turns into GDP. GTP is a triphosphate. It has 3 phosphates. DP has 2 phosphates. This goes from 2 to 1 to 2 to 3, and it's just sort of, it's routinely, it can happen. It's like a time reversal process. If we cut a leg on an arm, we can never have it again. But here, it goes back and forth. There's another book that was written, The Physics of Time Reversal by Robert Sachs. It says the higher the level of complexity, the more likely the time reversal process, because complex objects fall under the principle of Gestalt. They become Gestalts. It's more than the sum of its parts. Therefore, they can not go back, because that aspect that is more than its sum disappears. So this is what our personality is. It's more than the sum of its parts. This is a whole person is more than the sum of its parts. So we can only focus on molecular mechanisms if we want to look at time reversal. And there is, physicists now have just reversed time on the smaller scale by using a quantum computer. And you can see how it goes from order to degradation to time reversal and then back to order. And they have actually demonstrated that in physics to actually reverse time. We also have very interesting phenomena with resonance energy between proteins, kind of changing the process. You see how this one is not accepted fluorescence and this is accepted fluorescence. So there's sort of one protein can change another protein through resonance, which is a fascinating thing. So basically, if you had signals and you could insert signals through the resonance, through this energy transfer resonance, you could actually repair cells that have degraded due to wounds or age or whatever other reason. So now there is another issue, very important, is the timing within the cell. Every single cell in your body is controlled by its own circadian clock. Like we wake up and go to sleep, and that's a circadian rhythm. The cells have the same thing. So targeting that clock of the body, and you have, of course, clock genes and all these other kind of things that are involved in this timing process. The circadian genes in leukemia, for example, are faulty. And they have found that leukemia is a disorder that is associated to a disturbance in the circadian genes. The disruption of the circadian rhythms is followed by a disruption of cellular communications. So this is why one thing affects the other. So the timing of that if you wanted to sort of send signals into the body with very low energies under below thermal noise that can get through the cells, through the ion channels that open up, then you have to know about the timing. And there was a Nobel Prize in physiology and medicine that's awarded for the discoveries of molecular mechanisms controlling the circadian rhythm and how these molecular mechanisms kind of deliver the health status. Now, recent studies have also linked the skin circadian clock to psoriasis. And psoriasis is inflammation due to the disruption of signals delivered at the time schedule outside the required range of cellular circadian clocks. This is a study, we did several studies on psoriasis. This is Veronica Yap is one of our clinics that offers research. And you can see before and after here. This is again the same researcher before and after psoriasis on 10 treatments with this technology. Now, you want signaling that is emitted at specific times and that you have signals that can basically, through resonance, fix the signals inside the body. Basically, that's what you're looking at. And what is bioresonance? So you have like a sign that has sort of misses letters. That's a cell that cannot perform its function. And then through this resonance process, you basically reinstate the DNA sequence, and then the protein is creating the proper protein that will be functional versus dysfunctional. And this is how signal processing blueprints, signal plus analysis occurs. And there is it's a computerized method. It's extremely complicated that takes into consideration ultra low energies, target molecules prone to time reversal. You develop the blueprints of biosignals, establish the resonance, and then you have the control timing, and then you can sort of send them in to do the repair. And wound and scar healing involves a matrix of signals within a matrix of specific timing. So it's not just one signal, you have to have several signals. And the signals are basically composed by complex waveforms. It's about 4,000, each signal is about 4,000 waveforms, so 10,000 waveforms, depending on the particular signal. And they are joined together, these different frequencies are joined together to make one, because a resultant frequency, basically. And it has a specific timing, and it has a specific shape. This is a diabetic wound before and after 8 treatments. You can see complete healing with this technology. And this lady had very many problems. This is a liposuction wound, and you can see this is how it started. And then you can see that this is necrotic wound, which the necrotic wounds are very, very difficult to come back and to heal. And you can see how it became sort of alive again, and then it started, it gave a scar, of course, but this was several months. This is a postoperative skin cancer wound before and after 6 treatments. Again, this is all included in that paper that we published. Diabetic wound, 6 treatments during 1 year. Now, this all, we looked at after 1 year, all of these conditions, and the results persisted after 1 year, and some of them after 2 years. This is actually a varicella zoster virus, and that's the before, and this is after 2 treatments. This lady is 86 years old, and she had very good results, and again, that's the before and the after. You can see here, and you can see her lips after. All of these wounds are just completely healed. This is a traffic accident. This gentleman was told he will never be able to use his hands, and he had complete healing. He was young, however, he was like in his 30s. So the younger sometimes is much more easier to heal. Keloid scars and stretch marks are the result, again, of untimely, unbalanced processes, and also processes that continue despite the fact that they have fulfilled their function. And this is a before and after of a keloid scar. This is from Dr. Lampe's clinic. And this is actually only 1 treatment. We did, there's some, you can see some difference in the keloid. After 20 minutes, actually, you can see a big difference with this technology. This is stretch marks, only 1 treatment, 20 minutes, and you can immediately see that the pigmentation has gone down and the stretch marks have started repairing. This is a burn. It's a 3rd-degree burn, and sorry, 2nd-degree burn, I believe. And then after 4 treatments, you could see significant improvement, and of course, there was no pigmentation. The pigmentation started getting reduced. This is the before and after on facial rejuvenation. Also, it has an effect on hair. It increases hair growth. And like, what it is, can you actually unboil an egg? I mean, what am I talking about? And the truth is that, yes, there is a cell's toolbox for DNA repair, and unboiling that egg, basically making a protein being functional again, you can actually even start thinking about a cancer treatment. So you can unboil an egg by refolding natural proteins, which is exactly what this technology can do. And protein refolding occurs routinely inside the body, but however, through plasma resonance, if you insert a resonance, and there's different types of resonance, you can see a much faster result through different types of resonance in protein refolding, and this is the article that was written. And there is a measurement test. You can actually measure it. There is called the second virial coefficient, and when protein aggregation increases, that goes down. And when protein aggregation decreases, which means that you have a naturalization process, then there you have protein refolding that is successful. And with this, I would like to thank you again for your kind presentation attention. I would like to, if you need any information, it's a lot of information I present. If you need any more information about it, please email me at science@IELLIOS.com, IELLIOS.com. Thank you very much.